Okay, so in this video I'm going to demonstrate the method of how to test for disinfectants using the diffusion method. So we have here a PG plate, and this bottom of the PG plate has been divided into 5 segments because we're going to use 5 disinfectants to test. And we are testing E. coli and S. epidermidis. I already previously inoculated E. coli using a crosshatch pattern in this, and I have added the disc with the different disinfectants that we have here. Now the disinfectants that we have here are 1% bleach, a brand name with L, Mr. C, hydrogen peroxide, and 90% isopropanol. Now the way to inoculate the bacteria in order to test for the disinfectant efficacy is to use a cotton swab. You grab it by the wooden handle. You dab your cotton swab into the bacteria that you're going to homogenize by shaking it, because the bacteria by gravity tends to go into the bottom of the test tube. So you want to be able to grab as much bacteria as you can. And as with the aseptic technique, you just grab the opening of the tube and the cap, pass it twice through the Bunsen burner flame, dip your cotton swab into the culture, and then close your tube using the sterile technique. And then the crosshatch pattern, it's basically going sideways like this in your PG plate in a zigzag motion in one direction. Then you will rotate your plate and then go perpendicular to your previous streaks that you did. So you will do the same, close streaks, zigzag, and then you're done. And then you put your cotton swab in the beaker with the disinfectant, and then while you are going to add the disc, you meanwhile close your PG plate, sterilize quickly your tip of your tweezers, let it cool for a little bit, and then you have your disc here, which are filter paper discs, and then grab them with the tweezers, dip it into the corresponding disinfectant, and put it in the number where you have your labels. So this is number 1, this is number 2, 3, 4, and 5. So 1 disc, dip into the corresponding number for the disinfectant, and then you will be ready. So this is the disc for this 1% bleach solution. Open your PG plate here close to the sterile area, put it in the surface of your PG plate if it lets you, and then that's your first. Then just sterilize your loop again, your tweezers, the tip of your tweezers, let it cool, and then do the next. In the previous video I just did the demonstration of only 1, but I'm going to be doing it now for the 5, so you can see the entire procedure. So this is for number 2, which is here in this quadrant. And this one I had a little bit less trouble to put it in, just because I had less of the disc hole with the tweezers. And this is just a technical detail that sometimes you just learn as you go. So in the other part, I had it more way into the middle, the tip of the tweezer, and of course I cannot use my finger to slide it over the surface there. So I had to play around with it. Now these disinfectants, they have different components, and they have a different mode of action. Some of them, they damage. So now this is for this number 3, or for disinfectant number 3. So some of them, like in the case of isopropanol, since it's an alcohol, it will damage, let me put it in here, it will damage the plasma membrane. Alcohols dissolve lipids, and plasma membrane has lipids. So in bacteria, it will create pores within the plasma membrane of bacteria, and then you have leakage of the cytoplasm of the bacteria. So that's one mode of action of isopropanol, which I put for you there in the lab report. And you have to find out the mode of action of all the different types of disinfectant that we are using. So you need to know, in a way, what is the contents of each of these disinfectants. So that was then number 3, disinfectant number 3. Now let's do disinfectant number 4, which is this. It's hydrogen peroxide. So you put it there, and see how every time I open and close my Petri plate, this is just to prevent contamination, although it's close to the sterile area, but still you want to be able to work in a sterile area. And I'm trying to observe, since this is hydrogen peroxide, any reaction here with this disc in the surface, because later on we will be making something that's called the catalase test, which tests for the presence of an enzyme that is called catalase, where you add hydrogen peroxide into bacteria and you see bubbling of this bacteria, meaning that it can break down hydrogen peroxide into water and oxygen. And I'm kind of seeing faintly some bubbling in there. I might be wrong, but that's something to keep in mind for the catalase test. And now this is going to be the last disc, which is going to be for isopropanol. Now the other method suggested is the dilution test. It's a little bit more challenging. It takes more time, so we rather do this simple method, which is good. And we'll be doing this for the antibiotic test. So this is for isopropanol, and then I dip it in there and put it in my last segment in the Petri plate. And then one thing that you can do when you're putting your disc, by capillary action they will be stuck in, or get stuck into the surface, but you can press a little bit. Be careful not to apply so much pressure. And this is how your disc look like. So all you do, once you sterilize your tip of your loop, leave it in there, grab your 2 discs and see how the disc stays there. So the lid always goes down when we are incubating in the incubator. 37 degrees Celsius is body temperature, 24 hours. And then you will put it in here. Now before, let me explain you. So what we expect to see in the results, let me close the incubator, is a zone of inhibition of growth around the disc. So once the bacteria grows uniformly, you will have like kind of a whitish film in the surface of the agar. And if your disinfectant was effective against that bacteria, you will expect to see a clear area around the disc, meaning that the bacteria cannot grow because the disinfectant killed the bacteria. And in this case, you would call it effective. Now, if the bacteria grow exactly in the perimeter where you have the disc, this is a bacteria that is not killed by the disinfectant. In this case, you will call it resistant or non-effective. Now, if you have an area that is not too wide, but a clear area that is not too wide around the disc, that means that your bacteria was intermediately killed by the disinfectant. So you call it intermediate effective for the disinfectant. Now, at some point, we don't have the exact number of millimeters that you will take to call it that way, but this is just a school experiment to show the efficacy of the disinfectants. And then we grow a gram-positive, which is Staphylococcus epidermidis, and a gram-negative, which is E. coli. The differences in the cell wall can tell you why one might be more resistant to disinfectant than others. And this is something that you need to look in order to answer some of the questions that I have for you. This is all for this video. Bye-bye.